Now, thank you everyone for your attention. As Anna said, we are going to talk about plant isotopes, but applied to pastoralism, that it is what we are currently working in. So, first of all, I also wanted to thank you, the organization, for even if it's redundant, organizing uh, a meeting or a topic in this very, very interesting topic. And especially in my case, I'm kind of an alien in archaeology. I happen to be a veterinarian working in archaeology, so pastoralism and this topic is very special to me. So I will tell you what we are currently working in. So we wanted to explore animal diet in the past, and we have different approaches. Maybe, of course, you know probably all of them: dendral microwear, stable isotopes on teeth and bone collagen. Also, coprolites act as a kind of direct source. No, what you find on the feces is supposed to come from what animals have been eating. It's also been analyzed sediment from penning areas through fecal biomarkers, vegetal lipids from diet, but also micromorphology of those sediments, phytoliths, and something that we considered very interesting. Our point of start was shepherd knowledge. Why? Because unless we humans actively intervene on animal diet, animals don't have that cultural point of view of diet. So we suppose that what herds are eating now could have been eating in the past. So we planned maybe as Patricia said, we planned to explore the animal diet in the past through this traditional herding knowledge. So we are combining now what I'm telling you. Uh, the principal aims of this study are to reconstruct in this part of the Iberian Peninsula, but I will tell you later what the frame we started this work from. But now what I'm focusing today is to reconstruct this plant isotopic landscape of this part of the Iberian Peninsula, as I told you, Burgos, and cover different vegetation belts that could potentially act as a source of animal feed, to evaluate if there are significant differences between these zones we have selected, and to assess if this proxy could act as a useful tool to study present and past animal diet. So in a previous work, it all started with interviews with shepherds. We interviewed, I think it was 16 herders from 24 different locations from the Iberian Peninsula and Canary Islands. And since people from everywhere answered to this survey, we could gather data from probably most of the climate zones we have in the Iberian Peninsula. And with all that data, we selected the most reported plant species in each of those climates of the Iberian Peninsula. So that we are working on the paper, but it was already presented in a congress in 2024. And now we are focusing in a narrower frame, no, this part of the Iberian Peninsula, because we are working with in a site that is called El Portalón de Cueva Mayor in the Atapuerca sites. So we wanted to interpret what could be happening with these Neolithic and Bronze Age herders, and so we select, and the potential place what that they could be moving their herds that are currently nowadays the mostly the same routes they use, the very few herders that remain in this part. So in a range of less than, I think, less than 50 kilometers, we have these two very differentiated climatic and vegetal zones, and we wanted to know if there were significant differences between these plants and if plants could tell us something, isotopes from plants. So we parted from that survey and then we went to the field to collect those vegetal specimens. And for those, if someone, I am working more on the archaeobotanic point of view, and a partner of mine is more the archaeological. So I'm doing a very brief explanation of what we did. When you collected these plants, you have to take very, very much into account when you take it, because plant isotopes depend very much on climatic data, the coordinates, of course, the taxon, and the plant part you collect, because animals don't always eat the same part or the whole plant, no? They can be specific at some points. We collected the plants, we preserved them, and we prepared the plants for EI RMS analysis, which we are lucky to have that isotopic lab here in Burgos. We calibrated the results, and I'm presenting the first results. So what could be driving those differences, and what could be interesting for us to see and then to apply it to the archaeological record. So these are the potential sources of variation that could be acting in our data. So we plotted everything, and we didn't see anything at first. That was something that was kind of expected, because we are talking about C3 plant types, no? And well, we thought, well, what could be differentiating and what could be interesting for studying herds. We know that some animals prefer to eat trees, other shrubs, other herbs. So we tried to see if there were isotopic differences, significant isotopic differences between this type of plants. And as you see, there are no significant differences in isotopic variation, neither for carbon, neither for nitrogen. So we wanted to, we thought, well, what could be other use for our data? What could be interesting? And then the other question is that we are planning to say, no, that pastoral route in this part of the Iberian Peninsula, and we actually see that apart from that habitat or plant type, what is really significant in the isotopes, in the nitrogen isotopes, which are more variable, is the altitude. And we can see significant differences in regarding non-regarding the type of plant of these of the plants, and then we can apply it to, we are planning to apply it to animal data. So the two ecological places are very good differentiated, and I wanted to especially point out this type of plants, because they are always, in spite that they are different altitudes, they are always showing these low nitrogen values, and they happen to be heaths. And the heaths are a very important source of animal feed in these places, and they are also concentrated in this part of, I'm wait a second, because I have the panel of the Zoom panel and I don't know what I was pointing to. We have these heath zones and at this very part, so the potential pastoral route can be, or we propose that it can be differentiated. So maybe we are working on the data. I mean, it went out fresh from the oven 2 weeks ago. And what we can say is that based on our actual data, we can distinguish these ecological contexts, and that there is a clear ecological altitudinal signal in a very short range of space, that it is not dependent on the plant type, We need to check some special points before giving more conclusions. We need cross designs. We need to know if the same taxon eaten or potentially eaten by sheep show these different values or the same values in the different environments. Because if we are able to see this, we could use, at least in this part of the Iberian Peninsula, the nitrogen to combine it with the zooarchaeological data. And we also want to integrate this nitrogen, this plant nitrogen signal, with other mobility proxies such as sulfur isotopes or strontium isotopes. I think it is the work is currently being done by a partner here in the lab. And we want to ultimately integrate, as it has been made with humans, all the potential animal feed sources into vegetation mixing models to know more about the animal feed, both using um, uh, data from actual sheep from herds from this part again from of the province of Burgos, and then to apply it to the remains from the Neolithic and from the Bronze Age to see if we can know something more about animal diet, and always also taking into account that we are working with that we have to interpret all the data and all the zooarchaeological data with paleoclimatic and paleolandscape in paleolandscape terms, no? So I also wanted to thank again the herds that answered that survey, and thank you for your attention.